Welcome, everybody. I am Amy Shen, the Technical Account Manager of Sinobiological. Welcome to this webinar. In the next 30 to 40 minutes, I will walk you through the development of monoclonal antibody. We will briefly review the history of monoclonal antibody. I will use MAP to simplify it sometimes. Then proceed to discuss three technologies we use: Hybridoma Technology, Phage display technology, and single B-cell antibody technology. Let us take an overview of MAP development. It has been a long history since the discovery of antibodies. However, the MAP development goes back to 1975 when the first immortal hybridoma cells producing an antibody against sheep red blood cells was generated. MAP R&D platforms then keep optimizing, enabling the generation of a vast volume of antibody clones with surprising speed in the following years. The first antibody drug was approved by FDA in 1986. Since then, numerous MAP with multiple formats such as chimeric, humanized, bispecific have been applied to therapeutics. By today, more than 135 MAP drugs have been approved or under regulatory review for treating cancers, autoimmune diseases, viral infections, and etc. There is no doubt that more and more will be developed and approved for therapeutics in the future. Among the top 10 drugs by global sales, there are multiple MAPs for either for autoimmune disorders or for cancers. Again, proving the effects of MAPs in treating those diseases, also explaining why MAP development is such a hot topic, both in science or in business. MAP can be used as therapeutics, as mentioned in the previous slide, or as a reagent in diagnostics, like those in pregnancy test or COVID antigen rapid test. MAP are also widely used in biomedical research for qualitative or quantitative characterizing targets. Generally speaking, the technologies for MAP discovery had been developed for 4 generations. The first is the Hybridoma Technologies to develop immortal antibody-producing cells. The second is a generation of chimeric or humanized MAP by phage display. The third generation involves using single B-cell sorting techniques to obtain antibodies with native pairing. The most recent developed platform is using naive antibody library for antibody screening, which is most time-efficient way. It is not like smartphone that when the new generation comes out, the older one gets discontinued. All these platforms are used extensively in different scenarios. And today we will go through the basics of first 3 techniques: hybridoma, immunized phage display screening, and single B-cell sorting. There are mainly 4 steps in hybridoma generation: immunization, cell fusion, clone screening, and antibody production. Immunization involves injecting the target immunogen into animals. It could be a rat, mouse, or rabbit in tightly controlled schedule to maximize the immune response. Once the serum titer tells that the animal is ready, antibody-producing B cells will be harvested and fused to melanoma cells. Selections will be applied to remove unfused melanoma cells, and unfused B cells will die off after a couple of days, leaving only hybridoma cells in the culture. Then these cells will be subjected to screening by ELISA, flow cytometry, or cell-based assay, etc., to obtain the best clones. With the hybridoma cell line, antibody can be produced either by in vivo production through mouse ascites or by in vitro production through cell culture. Immunogens are not restricted to one type. From DNA to protein to cells, they can all trigger immune response with careful design of experiments. In fact, the choice of which format to immunize strongly influences the results. How the antibody generated be useful in different applications. For example, with the same target, antibody clone obtained from protein immunogen may have higher chance to work well in ELISA or flow cytometry than that obtained from peptide immunogen, since the latter may only recognize linear epitope that might not be accessible in the natural conformations. Besides the standard peptide and protein immunogen, we can also obtain antibodies using non-protein immunogens such as cells or DNA or viruses. Xenobiological built its legacy around recombinant production. Therefore, we prefer to use proteins as immunogens. Various production platforms can be used to generate the immunogens. However, many production is not very straightforward and needs troubleshooting for specific cases. Potential risks in the production includes and not restricted to low expression, aggregation, or degradation of the protein. The protein has very poor solubility, or it has high batch-to-batch variation. Okay, how should we tackle this? We can try different plasmids to help increasing production of the protein. Xenobiological has multiple proprietary vectors that have been optimized for recombinant production. Cultural conditions can also be optimized to solve problems such as low yield, degradation, aggregation, etc. There are a couple of case studies here are shown. The time of culture is correlated to the degree of degradation in this particular protein. So if we want high purity, non-degraded protein, we need to shorten the culture time. This is an example of baculovirus insect cell expression system. By optimizing the MOI, multiplicity of infection, a higher level of expression can be observed. Immunization strategies determines if a good serum titer can be reached, which is absolutely required for obtaining positive clones. Different immunization routes can be used. Among them, IP, meaning abdominal, and subcutaneous are the easiest, thus more widely used. For immunization schedules, you can stick to a routine immunization, which starts with first immunization, followed by sequential boosters, each time with standard amount of immunogens. Or you can do a rapid immunization with shorter time and a larger amount of immunogens. Adjuvants are also important in the experiments, especially for the target that exhibits low immunogenicity. A good choice of adjuvant can compensate that and increase the chance of obtaining positive clones. About cell fusions, there are two methods. One is Phage-mediated cell fusion. The mechanism is to use Phage, usually with an average molecular weight of 1500, to induce membrane rearrangement and cause the merge of two adjacent cells. The key factor for fusion efficiency are choice of the Phage, concentration of the chemicals and cells, incubation time, and temperature of fusion. The advantage of this method is low cost and done manually, thus does not require any special equipment. However, the disadvantage is that the efficiency is low and process can cause great damage to the cells. The other way is electrofusion. Electrofusion has gained popularity, and we at Xenobiological mainly uses this method. The cells are positioned on a point contact state, and membranes are rearranged and fused under stimulation of an electric pulse. Despite the need of an electrofusion instrument, the advantages of the electrofusion are very obvious. It allows for more control over conditions, higher efficiency of the cells, and as well as better growth properties following fusion. Moreover, it can be performed with a smaller number of cells. Here is some data showing the electrofusion versus the Phage fusion. You can see that for the same target, electrofusion can generate much higher number of positive clones and higher cell viability after the experiments, especially for the different targets, such as number 5. No positive clones were obtained from Phage fusion, but it is doable through electrofusion. After the fusion, extensive screening experiments are needed for two purposes. One is to isolate the clones that show high affinity, specificity, and yield. Second is to ensure the monoclonality. In this figure, an indirect ELISA was demonstrated. The target is coated on the solid support, or say ELISA plate. Antibody in the sample will recognize the target and bind to that. The interaction then will be detected by secondary antibody labeled with an enzyme that characterizes colorimetric reactions. The screening usually takes place in a couple of the rounds, at least 3 rounds, to make sure that the best clones are isolated for the later experiment. Besides ELISA, other screenings might be also used in different strategies to increase the success rate. For example, if we need to develop an antibody that will be used in flow cytometry, we can use flow cytometry in the titer test, the clone screening, and the subcloning to ensure that the final positive clone will be ideal for this application. At last, as mentioned, antibodies can be used either by in vivo production through ascites or by in vitro production through cell culture. Ascites production involves injection of hybridoma cells into mice and collect ascites for antibody purification. It is less controllable and has issues regarding animal welfare. The final product might also be contaminated by host proteins or even animal viruses. In vitro production involves culturing the hybridoma cells in flasks, bottles, or bioreactors. Currently, in vitro production has been optimized, in which serum-free culture and large-scale production are possible. So it is getting more and more favorable. Next, we will go through immunized phage display techniques. To start, we need to understand the biology of the phage. There are multiple types of phages can be used in phage display, such as T4, Lambda, and the M13 bacteriophage. M13, shown over here, has been used most extensively. It contains non-essential regions in its chromosome, so exogenous DNA can be introduced. Moreover, proteins are presented on the surface, making them accessible to the target of interaction. In the phage display technique, the feature of phage presenting a target protein outside is fully utilized. Here, the graph showing a recombinant phage displaying single-chain FV, aka scFv, which is the basic of a library. So let's talk about how we do the phage display. First, antibody sequence information is obtained from the B cells from spleen or peripheral blood or bone marrow, then constructed into single-chain FV. It is inserted into phagemid. A phagemid, like plasmid, contains antibiotic marker, gene coding the antibody G3P fusion protein and regions required for M13 chromosome replication. An M13 helper phage is usually used for packaging of the phagemid particles in E. coli. Once a library of phages displaying different scFv is constructed, it is ready for screening. Phage display screening is a high-throughput process including multiple rounds of 5 steps. Addition of the phage library into the antigen, phage with a single-chain FV binds to the antigen, washing off those non-bonders. Bound phages then are eluted by the enzymatic digestion using trypsin or by acid or base. The last, amplification of those binders. After a couple of the rounds, once the best clones are identified, the sequences of the single-chain FV will be cloned into expression vectors to make full-length antibodies. There are a couple of biopanning methods used in the screen. Immobilized method in which antigens are coded on the plates are most often used. However, in some cases, target might change conformation if directly coded on the plate. In this case, we would use in-solution biopanning to present epitopes better to the phage. There are also other ways of biopanning methods used widely. Consists of so many steps and so many rounds, phage display is not a simple experiment, and there are places can go wrong and can be optimized when dealing with special cases. We have established a mature strategy when it comes to troubleshooting for phage display. Changing the antigen coding conditions such as direct coding or biotinated antigen captured by immunized streptavidin will help the results. Careful selection of ELISA plates, blocking reagents, dilution gradient of library, coloring solutions, and playing concentrations are also ways to improve biopanning results. We have developed a long list of rabbit map by phage display. They have high affinity and sensitivity, specificity, and low background. Therefore, besides the regular application of Western blot, ELISA, flow cytometry, they are excellent reagents for IHC. And also they're ideal for map generation against mouse antigens. Besides the regular protein target, we also utilize phage display for anti-idiotypic antibody development. Anti-ID antibodies are antibodies that can recognize the antigen binding region of the antibody drug. They are inhibitory anti-IDAB that blocks the binding of an antibody drug to the target, and also non-inhibitory anti-ID that detects the total drug or the bound drug exclusively. Due to the application of anti-ID apps differ, it is of great importance to differentiate them during screening. One step is to test the serum titer and pick the one with desired binding performance. Another step is doing the phage display. Different screenings, such as competitive screening or complex specific screening, can be applied to obtain a clone with either inhibitory or non-inhibitory binding. These are results of our anti-ID app development towards anti-CD33 interleukin-17 reference antibodies. The resulting anti-ID clones showed no reactivity towards human IgG and isotype control, but great binding compared with the reference antibody, suggesting that those clones specifically target the idiotypic regions. Both inhibitory and non-inhibitory clones and paired antibody ELISAs are successfully obtained from the experiment. Next, we're going to talk about how Xenobiological apply phage display in nanobody development. Nanobodies are antibodies that have only heavy chains and have one constant region. So they only have CH2 and CH3, which make the whole molecule, is generally speaking, smaller than the regular IgG. They have also better solubility and higher penetration rate into tissue, thus has gained a lot of attention for therapeutic discovery. The development starts with blood collection from immunized llama or alpacas. VHH sequences then cloned into the phage mid to construct the library. Antigen-specific VHH are then identified by repeated rounds of binding, washing, and elution. At last, the best clones will be expressed as VHH or constructed into full length and characterize their purity and affinity and etc. Let us take a look at a case study regarding the development of antibody in the ADC, antibody drug conjugates. ADCs are intended to target and kill tumor cells. The antibody part contribute to the specificity and affinity, or sometimes induce therapeutic response. While the drug part, also called payload, it contributes to the cytotoxicity. They are linked by a linker that is stable in circulation and are able to release the payload at the target. After the antibody attach themselves to the surface of the cancer cells, they will be absorbed and internalized. The payload then kills the cancer cells or even take an effect in the tumor microenvironment. Here, let's talk about the target. Tumor-associated calcium signal transducer 2, aka TROP2 or epithelium glycoprotein 1. It is a carcinoma-associated antigen that is pretty popular right now as a drug target. It is a transmembrane protein that transduces an intracellular calcium signal and also acts as a cell surface receptor. The expression of TROP2 is very low in normal tissue. However, elevated expression has been observed in many types of cancers, making it a very good natural target for targeted cancer therapies. To obtain good clones for anti-TROP antibodies, we introduced cells into the screening process. Using flow cytometry, we can see that clones xenobiological developed has great specificity and affinity towards cancer cells, and almost no binding towards the epithelial cells. Regarding the internalization, the efficiency is comparable or even higher than the reference antibody. Using fluorescence, we're able to visualize the internalization of the antibody. Before the internalization, you can see that more fluorescence can be seen outside of the cell, perfectly outlining the cells, right? After 1 hour, you can see more fluorescence can be seen inside of the cells, indicating the successful internalization of the antibody. Also, it is reported that at an affinity towards target, a range of 1 to 10 nanomole, the efficiency of antibody internalization is the highest. The clones we developed all fall in this range, also proving that they can be a good candidates for ADCs. Next, we will talk about a single B-cell antibody development platform. First, let's review the immunology 101 for a minute. As shown in the illustration, VDJ recombination occurs in pro-B cells in the bone marrow, pre-BCR B cell receptors, then emerges on the surface in pre-B cells. Once the immature B cells travels to peripheral lymphoid tissues, the final phases of B cell maturation occur. The mature B cells will carry IgD and IgM on their surfaces, and those mature B cells bound to foreign antigens will get activated, then give rise to plasma cells for antibody secretion, and memory B cells who carry the B cell receptors. Single B cell sorting starts with collecting B cells from spleen or PBMC from immunized animals or infected patients. To enrich antigen-specific memory B cells, B cell receptors on the surface can be used to bind an antigen that is usually coupled to a fluorescence dye, followed by a sorting of the stained memory B cells by Phage. Then the B cells can be lysed directly to obtain RNA, or they can be put into culture. The supernatant can be used to isolate clones with best performance. Variable regions, they are cloned into vectors for recombinant productions of the antibody, and later they can be validated by various assays. The advantages of single B-cell platform is that the capacity of screening thousands and thousands of clones per round does make it more time-saving compared to hybridoma techniques. Second, natural cognate VH and VL pairing is preserved, making it has a higher chance to hit a clone with high affinity. Here are the flow cytometry results showing how we select the single B cells by flow cytometry from 2 species of animals, mouse and rabbit. For mouse, since there are panels fully established and available, we sort those CD19 plus, IgM minus, and IgG1 plus, and IgD minus cells. In addition, as I mentioned, antigen labeled with fluorescence is used to sort out those antigen-specific B cells. For rabbit, we use SSC and FSC to sort out lymphocytes first, because there aren't many established markers to use. And then we select those are IgM minus and IgG plus cells to, and those specific to antigens. Then antibody gene amplification is performed to clone the variable regions of heavy and light chains to expression vectors for recombinant productions. Let's take a look of case study. For antibodies used in immunotherapy involving T cells, there are two main categories: agonists and antagonists. Agonists, those are ones can activate the target, and antagonists can suppress or block the targets. OX40 and its ligand partner OX40L are expressed on activated CD4 and CD8 T cells. Stimulating OX40 has shown it to be a candidate for therapeutic immunization strategies for cancer and infectious disease. Agonist antibody for OX40 have to show high agonistic activities, as well as good affinity and specificity in cell or animal models. Here we screened thousands of B cells and isolated 300 ELISA positive clones. Among them, 10 clones showed lower blocking activity compared to reference antibodies. Then we examined the agonistic effect of the antibodies in the cell and found our antibodies work similarly or even better than the reference antibodies. Besides that, affinity of them have been shown to excel. IL-2 secretion by T cells also shows increased upon the treatment of our antibodies, suggesting that our clones can successfully trigger therapeutic responses and be a good candidates for antibody drugs. There's another case study we performed on TIGIT antibody discovery using single B cloning. PCR was done directly after sorting. We were able to obtain multiple clones that perform equally to or better than the reference antibody within a surprising short time, 26 days. To summarize, we have discussed 3 platforms of antibody development. They all have its own pros and cons. And Hybridoma Technology, the one with the longest history, produces antibodies with native and chain pairing, and antibodies undergo in vivo affinity maturation. However, the time and capacity are limiting factors for large volume screening. Also, genetic drift in the antibody coding region can cause batch-to-batch variation and instability of the cell lines. Phage display and single B sorting have solved the problem of long screening time and low capacity to a certain extent. Antibody sequences are obtained directly, thus production and batch-to-batch difference can be controlled more tightly. The disadvantages of phage display are that antibodies may not be native pairing. The library size is dependent on the E. coli transformation efficiency. And for single B cell sorting, the single cell sorting equipment is required, and thus there is higher cost usually. Besides, antibodies for B sorting are not available for all the species of the animals. At our Sinobiological, we can offer antibody development for anti-D, nanobody, bispecific antibodies, antagonists, agonists, and ADC antibodies, phosphospecific antibodies, and etc. We have rich experience and many successful cases for those projects. After antibodies are developed, we also have the capacity to characterize them by various cellular, biochemical, or biophysical assays. Sinobiological is one-stop reagent and crawl service provider. We have more than 6,500 catalog proteins, more than 14,000 antibodies, 32,000 cDNA products, and etc. Our crawl services cover recombinant expression for proteins or antibodies, antibody development, and customized assay development. Our product has been featured in many high impact factor journals and has spread their footprints in more than 90 countries. We truly hope that our effort can help your work and research and exert more and more beneficial influences on the scientific and human health field. Thank you.